Mutations is the next topic that we need to learn about. And it's a reasonably straightforward one after everything that we've just learned about gene expression and gene regulation. When we talk about mutations and looking at our objectives here, there's basically just two main categories we were going to look at. Number one is, have we had mutations in our actual genetic code? We know that our genetic code is made up of our nucleotide base sequences. And as a result of that, any changes to those nitrogenous bases within the nucleotides is a mutation. So do we have problems with our actual DNA code, or are there problems with things like chromosomes and the number of chromosomes and cells not replicating properly? That's our two main areas of mutations that we're looking at. If we have a look at our objectives here, we need to be able to identify how mutations in genes and chromosomes can result from errors in DNA replication, if we're talking about genes, and cell division, if we're talking about chromosome. We want to explain the term non-disjunction in reference to aneuploidy, which those words will make a whole lot more sense to you afterwards. We're going to use human karyotypes to be able to identify if there's been a mutation or an error in chromosome copy and be able to predict given data. So that's going to lead us to a little practical activity, analyzing human DNA data. And then lastly, we want to look at, well, we've got some mutations here, but how can mutations be passed on to the next generation, leading to offspring? Which leads us very well into our next topic, hereditary. So starting off, what is a mutation? You probably have all heard the term, but maybe are not 100% over it. A mutation, to be really simple, is a change in a gene or a chromosome relative to its original copy. It's an error in that genetic code. Mutations are really useful as well as also being potentially detrimental. They can produce completely new phenotypes, and that's how we get new genes that can be selected for evolution. That's how we get evolution occurring, is through mutations. So mutations can create completely new phenotypes, introducing new variation to the population, but they can also create detrimental phenotypes, which can have a negative effect. They tend to happen spontaneously during replication. So as your DNA is being copied, just random spontaneous errors can occur, which result in a mutation. But they can be also caused by a mutagen, radiation, toxic chemicals, or viruses. That brings me to one of our sort of main misconceptions when we think about, say, things like cancer or mutations. Whenever we look at a mutagen, or if we use the example of cancer, because cancer is an incredibly common mutation that I'm sure we're all aware of, when we think of something as being cancer causing, say smoking cigarettes or getting sunburned, that sort of thing, those factors, cigarettes, sun, UV radiation, are mutagens. They don't cause cancer. They just increase the likelihood that a mutation can occur. Let's say you've got a 1 in 2 billion chance of a mutation occurring each day. Smoking a cigarette might increase the likelihood of that mutation occurring to 1 in a million or something like that. So it doesn't cause cancer directly. It increases the likelihood that a mutation that causes cancer will occur. Hence why we have lots of people who will go through their lives smoking cigarettes, living into their 90s and never having a hint of cancer. And we're going to have people who've never smoked a cigarette in their life and they get cancer, despite that. So let's get into first topic is going to be about DNA mutations. So when we're looking at mutations within our genes, spontaneous mutations occur, as we said, can just occur randomly in the absence of mutagens, and usually are occurring when DNA is unwound for replication. So the DNA is unwound by helicase, and when that happens, it's no longer bonded and it's vulnerable, and as a result, we can have potential changes occurring. So When this occurs, we have a base generally swapped out for a new one. So a base might be changed. As you can see in this example, we're swapping a T for a C. And then when that strand is copied, we produce new DNA that is incorrect because it's incorrectly copied that, that error. When DNA polymerase goes along and checks for these errors, it can fix them. And in most cases, it does. In most cases, DNA polymerase does its job. It's copied. It's made all the new strand. It has corrected any errors, and everything's all good. And this is happening in your body multiple times a day, many of your cells. If the error isn't corrected, however, then that DNA or gene will remain mutated, and then it will be transcribed incorrectly into mRNA through protein synthesis, and then it will create the wrong protein in some cases. We can have a look here. We've got our original codon in the DNA, and we've got a potential change occurring. Okay, so when that change occurs, it can either be fixed or it can be mutated, as you can see in those two examples there. Well, a couple of different types of mutations, and this is one of the keys here. So when we're looking at a nucleotide base being changed or mutated, generally we're looking at a point mutation. So a point mutation is when a single base is changed. So it's called a SNRPS or SNPs, or single nucleotide polymorphism is the proper term. SNPs is how it's often pronounced. And what happens is one nucleotide, whether that be one that has an A, a T, a G, or a C, gets changed. The nucleotide gets swapped for another one with another base pair. And we can have three different outcomes of this. Number one is that it's a silent mutation. As we are aware, amino acids can be coded for by multiple different codons. And if the resulting change in that nucleotide is that it still codes for the same amino acid, then that gene will still get transcribed and translated and produce the same protein, so there's no problem. Okay, that's why it's called a silent mutation, because you wouldn't even know it's there. Nonsense mutation is when that mutation results in swapping one of those bases in the codon, and that causes a stop codon where an amino acid should be. Basically, you're cutting it short, as opposed to, say, having 200 amino acids for this protein. All of a sudden, at 73, there's an error which results in a stop codon, and then the protein doesn't get finished. The other one is a missense mutation, which results in a completely new amino acid being produced. You can see them illustrated quite well here. Okay, so a silent mutation, instead of being TTC, we're TTT, which means that the end result is we produce lysine still. So no harm to the end result. Nonsense, instead of a TTC, we end up with ATC. That ATC will result in a stop codon, which will mean that the proton will randomly and abruptly stop mid-strand. And then the missense, we could have, instead of TTC, we could have TCC or TGC, which would result in arginine or threonine being produced as opposed to lysine. And that is going to cause a completely new protein being made. And it could be a protein that does nothing, which means it's just trash and your body recycles it. Or it could be a protein that has a negative result, and that could be a big problem for your body. So these point mutations are actually fairly common, but our cells are, as we said before, the DNA polymerase is pretty good at proofreading and fixing this. It's very efficient. But if the change isn't corrected, then it becomes part of the base sequence and it will be copied throughout. So if I have a mutation in one of my skin cells, for example, that skin cell will later on in its life go through mitosis and be replicated and so on and so on. And now that mutation is part of my body. In cases of things like, say, pigmentation errors, not necessarily going into vitiligo, which is a genetic disease, but plenty of people have had errors previously where they will have, a little white patch pop up on their skin or a dark patch or something like that. That could be the result of a single mutation that's producing a different pigment instead. And that's not just going to hang around until that one skin cell dies. It's going to be there, forever because that's part of our DNA now. So we have a look here. Humans, we have a genome size of 3,100 million nucleotide pairs, and our mutation rate is 0.16 mutations per genome per cell. You can see here quite interestingly, the bigger your genome is, the more mutations the mutation rate is higher, which is quite an interesting phenomena that we see in nature. We'll talk about that more in evolution. So point mutation is pretty straightforward. We just, do the old switcheroo with one of our nucleotide bases, and the end result is we end up with either no change, which is a silent mutation, we end up with a stop codon, which is a nonsense mutation, or we end up with a completely new amino acid and a dysfunctional protein, which is a missense mutation. So they were our substitution mutations overall. Then we've got frame shift mutations. Frame shift mutations shift the way that it gets read. So we know that our DNA is read in terms of 3 bases at a time, which is our codon. A frame shift mutation is a mutation that changes that reading frame. So by either inserting or deleting. So we're not just switching one out, we're either adding a whole new nucleotide or getting rid of one entirely. So insertion is when we add one or more bases. Deletion is when we remove one or more bases. And because we do read our genetic code in codon, so it's 3 bases at a time, inserting or deleting is going to change the way that it gets read. So you might have had, most of the right nucleotide and codons in there, but now we've shifted it. Looking at this example here, we've got our DNA TAC, TAT, GCC, TTA. Okay, if we remove that T, instead of producing methionine, isoleucine, arginine, and I can't remember what ASN is, then it all shifts. By just moving that T, we've now got TAC as our first one, yeah, but instead of TAT as our second codon, as you can see there, we end up with ATG, which codes for tyrosine. And because we've got ATG, the G from the next codon, which should have been GCC, is not there. So instead we read CCT. And then at the end, we've got TA, which is nothing. So not only by deleting that are we producing a shorter polypeptide, but it's also incorrect. You can see our errors here in terms of insertion, deletion, or substitution. Substitution, we already talked about, pretty straightforward. It's just a switcheroo. Insertion, it's going to completely change the reading frame and add an extra base, giving us 10 overall. Deletion, we're removing one, it gives us 8, and that's going to change things also. So it's a big problem there. So we already talked about mutagens briefly, but effectively, and for all you guys need to know, is that we have both chemical and physical mutagens. And as I said, the really important misconception to get out of your head is that mutations are not caused by the mutagen. They're not induced by the mutagen. The mutagen only increases the rate of mutation. Recently, the Australian Cancer Council published some information where they said that we should no longer be putting ham in children's lunchboxes because the nitrates in the ham can cause stomach and colon cancer. Now, that doesn't mean if you eat ham, it's going to give you cancer. It just means that by eating that ham, it slightly increases the rate at which mutations occur, which could result in that cancer. And why stomach and colon? Because that's where ham interacts with your body. We have chemical or physical mutagens. Chemical mutagens are anything with chemicals in them. So easy example is cigarettes, drugs, alcohol. Then we've got food and other chemicals as well, poisons, etc. Physical mutagens are our radiation, so UV, any sort of basically electromagnetic radiation, UV, X rays, gamma radiation. All of that is a physical mutagen, which could result in mutations that can cause cancer. Or, if you believe Marvel, gamma radiation will turn you into the Incredible Hulk. As well as this, we can have mutations induced by infectious agents as well, bacteria, pathogens, basically. So chemical mutagens, we have huge variety of them, and a lot of them act in a different way. So this is a list of some common ones here, and they probably make absolutely no sense to you. You probably have no idea what they mean, but you can see acridine orange, bases are added or removed. So we're looking at that in frameshift mutation. For 2-aminopurine and 5-bromouracil, nucleotide substitution. So you have that switching. Polycyclic aromatic hydrocarbons cross-link with adenine and guanine, which stops any repair of the DNA, etc. You don't need to remember all of these, just need to know chemical mutagens can cause mutations. And physical mutagens, such as ionizing radiation, which I said before, electromagnetic radiation, basically the radiation, the energy in that radiation, those particles of radiation, is so strong that it can break the chemical bonds in our molecule, such as DNA. So it removes tightly bound electrons, causing the atoms to become charged, and that can affect the DNA quite significantly. When we look at UV radiation, it can cause base mutations, and that's what's going to give you things like cancer, for example. We've got our X-rays, which can cause deletions, so frameshift mutations of A's or G's, and that seriously messes up your DNA. And then nuclear radiation, which is bad, causes double-strand breaks. Double-strand breaks can be huge issues because they can result in large sections of your chromosomes being completely messed up. And if you have too many of these, the cells die, and from there, basically, you die. All right, that's our DNA mutation. So I'm going to end it there. In the next video, we'll look at aneuploidy and what happens when you have the wrong number of chromosomes.